Hello, welcome to the CBCAN. I'm Jim Audia, Executive Director of CBC. I think we'll go ahead and get started. I've been trying to be a little bit liberal on the time here because we're in a different location than traditionally, and also we've had some traffic challenges associated with the weather. But I think it's time to go ahead and get started. So one of the things that we do traditionally at the start of each of these CBCAN meetings is go around the room and let everyone introduce themselves and their affiliation and talk about what their role is within the community. And so most of you probably know I'm Jim Audia, head up the CBC. I come from a background of drug discovery, both in biotech and pharma, and excited to be here. And so why don't we go ahead and do our introductions, and I think we can pass microphone around and just let people, again, tell us a little bit about yourself, because part of the reason for being here is to network and get to know each other.
Hello? Okay. So I'm Yola Glotzer. I'm with the CBC. I'm CBC Executive Communications Director. Not quite yet.
I'm Phil Hockberger. I'm from Northwestern University. I'm the Associate Vice President for Research.
Yeah, my name is Xiaoping Du. I'm a professor in the Department of Pharmacology at the University of Illinois at Chicago. I work on the basic, mostly a cardiovascular field, and work on also translationally. We work on the anti-thrombotic drugs. Where try to develop a new type of anti-thrombotic drugs that doesn't cause bleeding.
Hello, Rich Minshall from UIC. I'm a professor in pharmacology. Work actually closely with Xiaoping on one project related to thrombosis, and I'm also now the director of the Research Resources Center, RRC, and I'll have a chance to tell you a little bit about our focus areas related to drug discovery here pretty soon.
Hello, my name is Manel Benaissa, and I work at UIC2, College of Pharmacy. I'm a research assistant professor. I work in the US Center for Drug Discovery in the Thatcher Group, and I mainly focus in neurodegeneration. I'm responsible for bioassay development and testing in vivo and in vitro.
Hi, I'm Steve DiMagno. I'm a professor of medicinal chemistry at UIC, and my specialty is really preparing radiolabeled compounds for imaging and therapy.
Hello, I'm Yuan Zong with Surat Therapeutics. We spun out the University of Chicago in 2016 and incubating at the Polsky Incubator. And we're developing a new medication for migraine that can be delivered intranasally.
Hi, my name is Mike Flavin. I'm with Flavin Ventures, and I've been involved with the startup and development of several drug discovery companies here in the Chicago biotech ecosystem over the last 30 years.
Hello, my name's Joe Austin, and I am the Facility Director of Electron Microscopy Core at the University of Chicago.
Hi, I'm Vivian Prindle. I'm with Northwestern, and I'm a project manager in the Lurie Cancer Center.
Hi, my name is Irawati Candela. People call me Anki. I work at Developmental Therapeutic Core. I'm helping professors, PIs, postdoc, graduate students with preclinical studies. So we're doing a project with pharmacokinetics, efficacy study, toxicology studies, and some of them are imaging and working with histology core, as well as a genomic and some other core. Thank you.
Hello, I'm Karen Snap. I'm the Associate Director of the Chicago Biomedical Consortium. I work on a lot of staff projects and also in charge of several of the award programs.
Hi, I'm Stephanie Colonna. I'm assistant professor at UIC in the Department of Chemistry. My group is primarily a mass spectrometry lab. We do proteomics and lipidomics to understand neurodegenerative diseases, and we work on a rare disease called Niemann-Pick type C.
Hi, I'm Laura Sanchez. I'm an assistant professor at the University of Illinois at Chicago in the Department of Medicinal Chemistry and Pharmacology. And my lab also uses mass spectrometry, but we're using metabolomics to look at how cells and microbes communicate with one another in and on things.
Hi, everyone. I'm Brian Kay, a faculty member at UIC. My research group is interested in using phage display for protein evolution and engineering experiments. I'm also the CEO of a startup here in Chicago.
Hi, I'm Karina Gicharan. I'm with the CBC project coordinator stationed at UIC.
Hi, I'm Connie Lee. I'm the Associate Dean for Basic Science at the University of Chicago, and I'll be giving a short overview of our course.
Thanks. I'm Lucy Godley from the University of Chicago. I'm a physician scientist. I study cancer biology and see patients with blood disorders, and I'm also the scientific director for the CBC from the University of Chicago.
I'm Julian Salway, Dean for Translational Medicine at University of Chicago and one of the PIs of our CTSA.
Hi, I'm Dimitra Georgianopoulou. I'm Director of Commercialization from the Innovation and New Ventures Office at Northwestern University.
My name is Chihao Luan. I'm the scientific director of the High Throughput Analysis Laboratory at Northwestern University. We provide high throughput solutions for small molecule screening and biological screenings.
Hi, I'm Chad Haney. I should be the poster boy for CBC. I got my PhD at UIC, did my postdoc at University of Chicago, and now I'm a research professor at Northwestern. I run the Center for Advanced Molecular Imaging.
Hello, my name is David Leclerc. I run the flow stoichiometry core facility at the University of Chicago.
My name is CBCAN June. I am the technical director of the Cellular Screening Center. We do high throughput screening on small molecule and siRNA.
Mike Johnson at UIC, mostly in the Center for Biomolecular Sciences. I'm also involved with a couple of startups and work mostly on antibiotic discovery.
You don't get away without introducing yourself, Nancy.
I'm the one who got lost. So I'm Nancy Harvey with the Polsky Center at the University of Chicago. Pleasure to be here.
Hi, I'm Terry Frederigo from Northwestern, and it stopped raining outside. Yay!
Yeah, all right.
I'm Nancy Terrell, Associate Director of Translational Activities, and I welcome everyone here. Thanks for making the effort despite this rotten weather.
Yep, so anyhow, I appreciate all of you hardy folks for showing up despite the traffic difficulties and the weather, and actually excited about this program that we have this evening. You know, I do think it's an exciting time to be here in Chicago and trying to do biomedical translational research. There's clearly a buzz about the city, and you know, a couple of recent occurrences have, I think, further added to the excitement here. Those of you, might have seen Mike Flavin at the Bruce Rauner Rahm Emanuel press conference, kicking off the DPI, the Discovery Partners Institute down south. I think that's going to be an exciting addition to the community here in Chicago, and I was really pleased to see an elevated role for UIC within DPI, and I think this is going to bring additional resources to the translational efforts in the community. Last week as well, we kicked off Lakeside Discovery, a collaboration, a partnership between Northwestern and Deerfield Management, which is going to bring a fair amount of investment capital to the community, again, to do translational research. And so, I think this is really exciting, and I think it's pointing to the fact that Chicago is an increasing center for biomedical research. And so, why is that the case? Why is investment money coming to Chicago? Well, I think there are a couple of reasons for that. Firstly, I think we have just, fantastic research universities here in Chicago. We have Northwestern, University of Chicago, UIC, and then in that extended community, there are other research institutions as well. Secondly, I think those universities have just fantastic faculty, and so, having brilliant, innovative scientists like yourselves and your colleagues, I think create a number of interesting possibilities. I think thirdly, I think we have an increasing level of infrastructure here within these universities and within the community. I think CBC has played a role in much of this. I think we've helped to provide guidance, provide funding, provide dollars to recruit faculty and to retain them, and to invest in infrastructure. And I think importantly, what we've been able to do is to increase the level of collaboration and access to that infrastructure. And so today's program is really focused on a component of that infrastructure. And so I'm really excited to both learn more about it, but also to stimulate a discussion around it. And so I'm going to turn the podium over to Nancy Terrell, who's going to introduce the speakers, but I seem to be missing Nancy. Anyhow, I think we're going through a few audiovisual gymnastics as well here. See, this is where my training as a lector came in, Yola, where I could stand up here and talk with all this distraction going on without me, without even noticing it, right?
Yeah.
So we're... Bear with us for just a moment. Okay, I'm going to turn the program over to Nancy, who's going to introduce our panelists.
Thanks, Jim. So welcome to the event this evening. Again, we appreciate you all coming out to this important and interesting topic of the core facilities between the three CBC institutions. Those include University of Illinois at Chicago, University of Chicago, and Northwestern University. So we were fortunate to engage the core directors from each of those universities, and they will be speaking to you tonight featuring different cores that would be relevant to the drug discovery area. I was shocked to learn from Phil Hockberger that the universities have, I think it's 60 at Northwestern, 60 cores, am I right? Somewhere around there. I thought it might be 10. I was surprised that there are, it's so full of different cores that would be available to the faculty members and researchers to utilize, especially through the open agreement that we have between the three, the Open Access Initiative. So Phil will be talking a little bit about that. We will tonight feature Phil Hockberger will get us started. You're welcome to come up to the panel. Phil represents the Northwestern University research area. He's Associate Vice President for Research. And then we welcome Connie Lee, who is kind of filling in for George Langan, and we appreciate that, Connie. George is really the head of the Research Resources Center at University of Chicago, but Connie has brought along with her 3 individuals who are experts in their area, and we can turn to them if we have questions about that. So, and then next, Rich Minshall is representing University of Illinois at Chicago, and will be covering a couple of unique areas that the UIC offers as part of their core. So we welcome you and look forward to getting started. Thanks, Phil.
Good afternoon. I'm glad you could make it. So the 3 speakers are going to cover slightly different aspects of the topic today, and I think we've got a nice tag team plan for you. I'm going to start off by talking a little bit about the Open Access Initiative, which is a partnership between the 3 schools in the core facility space. And then I'll transition to a little bit about what Northwestern is doing in their drug discovery pipeline and how the cores fit into it. Then Connie is going to talk about the cores in particular, take a little deeper dive at the University of Chicago into cores that participate in their drug discovery efforts. And then Rich is going to come back and talk a little bit about the cores as well there that are contributing, but go into some of their ecosystem and a couple of the centers that have become important players in that pipeline at UIC. What's important for this audience to understand is that the strategy that all 3 universities are taking in not only the drug discovery space, but research in general, is to do this in partnership. Now, that partnership is not just core facilities. That partnership is across various research programs and centers. Julian and I are both involved in our CTSAs. I'm an associate director of ours. He is one of the PIs of the one at University of Chicago. We have members from UIC also on that advisory panel. And there are a lot of activities in the clinical and translational science area that we're doing together in partnership. And there's lots of reasons for doing this, not the least of which is we all share the same geographical areas, and we want to be able to work together, not in competition with each other, but in partnership to bring better healthcare to the entire metropolitan area. So the core facilities started with a memorandum of understanding. This was an initiative that, in fact, I pioneered back in 2010 with the help of the CBC. The CBC funded the feasibility study with Huron Consulting, and that study was intended to look at ways in which we could work together, core facilities being one of the ways that we could work together. And Huron then interviewed senior leadership, they interviewed faculty, they interviewed cores, they interviewed scientists at all 3 institutions, asking the question, how would you feel about having access to facilities and faculty at the other schools? And the message that came back loud and clear as a result of that study was that the researchers would welcome this opportunity. So that report was in 2010. In 2011 and 2012, we started meeting with each other, the core leaders. And by 2014, we had a memorandum signed by all 3 provosts that began with a fairly modest ambition. The modest ambition was, how about we just drop the indirect charge for using each other's facilities? That indirect charge was like a surcharge that we charge everyone else, an administrative overhead for using our facilities. But if we were each using each other's facility, that's kind of silly to be charging each other a tax when, in fact, we would be wasting dollars that could be spent on research on this indirect charge. So the provosts all agreed we would be modest in our ambitions and just start this project 2014 by dropping it. Well, it's turned out to be a very successful program. We have an advisory committee made up of 5 faculty from each of the 3 schools. They meet every fall, and They met in 15 and 16, and then the one we met in 17, and Jim attended this one, and Karen attended this one. When we met in 17, we decided instead of renewing it every year, which is what the memorandum called for, we were now going to renew it every 3 years. And the advantage of that is that the faculty can now write into their grants, I have access to these facilities through this program for the next 3 years. So to me, that is a testament to the success of this program and the value it is bringing to the researchers. So that was, the core facilities was one of the recommendations of this feasibility study. The other recommendations were joint workshops and teaching opportunities, which we've done through the CBC, joint grant applications. In just the last few years, we've had 9 grant applications from our partner institutions. 8 of the 9 have been funded. The only one not funded was the cryo-EM. We still can't figure out what happened. But nevertheless, Joe, we'll figure it out someday what happened. But we've had tremendous success in that. And then joint networking opportunities. And one of the events that's coming up now on September 7th is for the core facilities personnel, the staff and the directors to meet at CG Life in downtown Chicago for a social event. Again, part of trying to build community with each other. So if you go to the CBC website, you can learn more about the Open Access Initiative. There are links here to how to get involved for the faculty. There's also, at the bottom down there where the arrow is, a link to all 3 websites for the universities of their individual core facilities. If we were to click on the Northwestern one, you would see that there are all the facilities listed. This is just the page on the Chicago. There's also a page for Evanston. I've arrowed the ones that are involved in drug discovery. So Center for Translational Imaging are the MRI facilities. There's 5 human magnets, 1 animal magnet in Chicago, 2 animal magnets in Evanston. In fact, Chad, who's in the back here, runs those at what's acronym is CAMI here on the Evanston campus. Then we have the Clinical Pharm Center, the Matthews Center for Cell Therapy, the Peptide Synthesis, Structural Biology. Proteomics is not on the list. I need to get it on there. That's in Chicago. Those are all cores that are actively involved in the various drug discovery pathways. On the Evanston campus, we have the high throughput. Chi Hao is here from that, the HTAL. DTC, that's Anki's, she's here today. Diagnostic Therapeutic Core. I mentioned CAMI. Center for Advanced Molecular Imaging, and then RPPC, the Recombinant Protein Production Core, all of those. And there are many more cores, as Nancy said, not 60, we're closer to 50. Oh, and Terry Frederigo, who's here from ChemCore, which is also should be on here. So we have a lot of cores on both campuses that are involved in drug discovery. Now, if you go to INVO, which is our tech transfer website, there are a number of resources here. On the left-hand side of the screen, you'll see there's a guide, a startup guide. Again, this is for the faculty who are interested in thinking about moving into this space. The flow chart here on the right-hand side show a number of different processes, but I want to focus your attention on just the information that's available for the faculty from any school. If you want to come and just learn about this, this is a website that will walk you through it. Jim mentioned the Deerfield partnership that was just announced with Northwestern. Jim is, in fact, on the steering committee, and that's going to be part of the, I'll mention that shortly in the flow chart of how they fit into this, this pathway. But that was just announced at the end of May. We had a big kickoff just, I don't know, a few days ago that we were all at. This is very exciting because this is really an opportunity to move projects way forward. There are a number of gaps. In fact, the take-home message from my talk is going to be the gaps that still exist, and this filled one of those gaps. So this is the flow chart here, developed by Carl Scheidt. On the far left in the red box, this is the early discovery, and this is where the accelerator program that the CBC funds, this is the kind of work that we're talking, very early, basic research, but with potential. And the awards here are the CBC awards, the Catalyst and Accelerator. And notice the question mark down at the bottom, because one of the gaps that still exists are the people who can help faculty working in this space know about the facilities that are available, know about how to move things through the process, what's going to be needed, even to help educate them in the preclinical and the external areas here to the right. The Northwestern has developed this ecosystem to help move projects from the early through the preclinical into the external. And notice here under the external, Lakeside Discovery. Now, Lakeside Discovery can help at the end, but they can also help in the preclinical, as do NIH grants for SBIRs and STTRs. They are all part of this ecosystem. Northwestern has, again, emphasized that there are different steps that faculty need to move their projects through. And so we can get into this in more detail, the question and answer. This is the last slide. These are the gaps. One of the reasons I agreed with to come and give this talk for Nancy was because there are still gaps here, gaps that we can fill together. I got this primarily from Carl Scheid, who runs our new cures development, and a faculty member in chemistry. The compound stability, once you have a compound, it works maybe in culture or some in vitro system, once it goes into the animal, these compounds sometimes are broken down right away, and so they're not stable, and this kills the project right off the bat, and you can't move forward. And so we need to figure out how we can, as Carl's fond of saying, which is fail quickly. If you've got a great idea, let's get it into the pipeline and find out right off the bat by doing the key experiments, is there any hope that this is going to be useful? Potency, at what concentration? If it's going to take large amounts of this compound, which has potential toxicity, it's a non-starter. Chemical libraries in our high throughput screening, there's still fairly limited libraries, so we're not really screening all the potentials. Libraries are expensive. And verifiability, it's going to become increasingly important that you outsource some of this work to an independent lab who can verify this, independent investigators. And it's because as investigators, we look through rose-colored glasses. Anything that looks positive looks good to us. But I think in the commercial space, the rigor is, the bar is much higher, and verification is going to be a part of this. In the synthesis, radiochemistry, Stephen, where are you? There you are. This is an area that Northwestern would really like to move into. University of Chicago is moving into it. UIC now is partnering with them. And I think we would like to have discussions about this because, as Chad has explained to me, we really don't have a way. What we're talking about, my friends, is if you have a compound and it works, that's great. Usually it's in a culture or some model biosystem, but the moment you put it in an animal, we can't see it anymore unless there's a way to tag it. So we need somebody who can take the compound of interest and put a tag on it in some way that we can follow it with our animal imaging methods while it's in the animal. Somebody's got to have that expertise like Stephen to help us. So there's an opportunity there for filling that gap. And then finally, mentors and navigators. Mentors are people who've been through it before, who know this space and can help mentor our faculty. Navigators are people whose job it is to help you move your project through the different cores. Again, all in the early discovery area. All right, so that's it for me. I'm going to pass it over to Connie now, who's going to come tell us about the University of Chicago's cores in the drug discovery area.
All right, thank you, Nancy. Thank you, Phil. I'm happy to be here from the University of Chicago. And as Nancy indicated, I'm actually not overseeing the cores. So George Langen is overseeing our cores, and he's traveling this week. So he had asked me to fill in, but I'm happy to be here. And as mentioned already, we have 3 of our core directors here as well, Joe, David, and Sequan, who will be able to answer more detailed questions. So our cores, we don't have 50, but depending on how you count them, it's somewhere in the 25 to 30 range of cores. Here we have it divided into the different areas. And These are all overseen by a Office of Shared Research Facilities, which we call OSERF, so which provides, as probably the other institutions have as well, some general oversight, financial oversight, billing, web, IT, HR type services for the course, so they don't have to all do this individually. And I will also mention, I should have probably prepared a slide for this, we are in the process of also launching, we got a large philanthropic gift from the Duchossois family, so we are in the process of launching the Duchossois Family Institute. And as part of that gift, the idea was that we would start some new technology platforms. So please stay tuned for that. But two of the platforms that were proposed were medicinal chemistry and metabolomics. So that would be relevant for this office. We're currently searching for a faculty director, and we thought the faculty director will also have some say as to what technology platforms will get developed, but we're hopeful that those two will be part of the mission. So the four cores that George and I decided to focus on for this meeting, two of them somewhat more related to drug discovery and development, and two others that we wanted to feature some new equipment or cores that have actually received support from CBC. So the four that I'll talk about briefly are the cellular screening core, advanced electron microscopy, cytometry, and the cyclotron. And I've just highlighted those in red. So the Cellular Screening Center, as Phil already mentioned, we have this. This I learned when looking it up also received support from CBC very early on. So I believe the other universities here also have a cellular screening as well, but we're, I guess, one of the only ones that has it in a clean room facility. Has a F3 robotic arm that allows high throughput, walk-away type screens, and we do both genetic screens and chemical screens. And as Phil already mentioned, we do have several libraries for the chemical screens. We're looking to perhaps improve the libraries at this time, but CBCAN can also answer any more specific questions you have. So cytometry and antibody technology. So we wanted to mention this just because we also have a new CyTOF machine in this core. So basically, CyTOF is a mass cytometry, so that basically combines mass spec and some of the features of flow cytometry. And one of the advantages, it allows about 37 different types of tags compared to flow cytometry. I believe there can be 17, 18 different tags detected, and so that's a real advantage. And it also can extract more information from more limited sample size. And David is here today and can answer any questions you have about that. So the cyclotron, this is relatively new. I think it's been in place almost 2 years now. This is basically a type of particle accelerator, and so you can imagine it was sort of a operational feat to get this into place. So Rich wanted me to make sure I showed a picture of it, and to just talk about some of the logistics. You need the certain thickness of walls. It weighs 27 tons. So obviously this was really difficult to get into place, but it is operational. And this is a list that so Richard Freifelder was not able to be here today. He's actually taken off for a nuclear medicine conference. So he's our program manager for this facility. He wanted me to indicate, they have made several different types of labeled isotopes. These, I don't know the half-lives, but they all have varying half-lives. We've also set aside funding to create a room for those doing mice work, so it can be right next to the cyclotron, because some of the things, again, that you make are very short-lived, and so we are still in the process of setting that room up, but that will allow you to work with some of these very short half-life labels in right next to the cyclotron. And lastly, I'd like to talk about our advanced electron microscopy core, and Joe Austin is here and can answer more specific questions. But this is, obviously, as most of you know, there's been, this great, technological advances in electron microscopy over the last few years, especially at the cryo electron microscopy range, which basically allows it to get to single particle atomic, single angstrom resolution. And so electron microscopy can cover this huge range of power. And so this obviously takes a lot of technology to cover such a huge range. And so I wanted to mention this as well because we got into cryo-electron microscopy about 3 years ago, I think, again with the generous support from the CBC that allowed us to buy our first cryo-EM, a Talos machine. But we have several faculty at University of Chicago who really thought we also needed to get into the next level, so get higher resolution and get a Titan Krios. And so the University of Chicago, we've just put in orders for these 3 new pieces of equipment. So one is the Titan Krios, and then Aprio for the more volumetric EM, and also to be able to do tomography as well. So this is the slide that Joe prepared. So basically we can now, after these new instruments arrive, which again, for the Titan Krios, unfortunately there's almost a 1-year wait time to get the machines. So it's gonna be a few more months yet before we get that. But once all these new machines are in place, we'll be able to basically cover the full spectrum. So from doing the volumetric EM, basically reconstructing a synapse, all the way down to single particle analysis with the Titan Krios. And I think that was all I had, so we're happy to answer questions later. And Rich.
Yes. Okay. Hello, everyone. I have to first preface this by saying all I know I've learned from Phil. So I inherited a role as director when my predecessor, Bill Hendrickson, retired. And Bill had worked with me for about a year or so, kind of grooming me for this spot. He thought I would be the right kind of person. I can just tell you that it really is a culture of us in this environment that are collaborators and or enablers, that our job is to provide high-level service and means to an ends for faculty and students. And so I'm listing here now what I think is an important aspect that I'm trying to do at UIC. First, I had to organize, I think, a little bit. We were a little bit chaotic, but the critical element in this list is the let's say the key the key cores. The key cores that basically all institutions need or have if you're a decent-sized medical center, and the list of those is familiar to everyone. And then perhaps some specialty areas. We happen to have a decent population of cardiovascular scientists. We have a decent population of neuroscience group people. So, what are the cores that you really do need? Do you have the right PhD level experts in that arena that can help facilitate the training and learning on that new instrument or that method? And then do you have faculty who are really engaged? And I think this is really I'm finding, one, very helpful, and two, critically important as you want to make any kind of step to campus level leadership to say we need to improve this or that faculty who are engaged are extremely important to be able to do that. I've listed key people here. Many of you guys know these folks, and a couple of them are here today with, as part of this effort. Stephanie Colonna, who introduced herself, but new faculty member in chemistry, but really an expert in mass spec. I need this kind of expertise because I am not an expert. I need the core director to really be able to latch on to somebody for guidance. Laura Sanchez, sitting right next to us, also one of these folks that are very generous of their time. Mike Johnson's here, Greg Thatcher's here. These are critical faculty that I think the cores can't function without that level of support. So just to highlight what I think are areas of expertise at UIC in the realm of drug discovery, drug development, have 4 or 5 slides kind of tagging these. There have been long-standing efforts. UIC has had a Center for Structural Biology, largely driven by groups in medicinal chemistry and biochemistry. They had crystallographers. They had people like Mike Johnson doing drug discovery and SPR, kind of biocore-related binding studies. How do you take this to the next level? What can you glean from small molecule crystals out of argon versus high-field NMR? And so this kind of approach has been pretty successful at UIC. I would suggest that there has been the onus of an effort in drug discovery based upon some facilities and key faculty in this area. And I think that is probably going to do nothing but expand in the near future. So we're trying our best, at least from my level and through the Vice Chancellor's office, to try to maintain and build our capacities through these high field NMRs, and then to stay current with technology in terms of efficiency of doing these binding studies. So the next technology is now much better. We're trying to upgrade some instruments to make this more high throughput. Also measure high throughput screening, which is something that we have a core for, this robotic small molecule screen based upon this TCAN robot and plate readers that allow you to identify if you have a very robust targeted screen assay that you could now go through this compound library. And being able to do this with some guidance, right? I'm approaching this now as a faculty member. It's really critical. I actually have a postdoc who has some background in this area, and I have to rely on people like Greg Thatcher to guide me through this. Otherwise, I'm just kind of spinning my wheels. So I have another slide. I'm going to talk a little bit about Greg's approach. We have a mass spec facility, which by and large started because of the CBC, that there was a gift, that there was investment in proteomics instrumentation. And this has rapidly evolved. We've spent a little bit of time and money trying to reposition the facility, to put more support and guidance in the form of faculty associated with the facility, and now to try to add instruments that are either less than par or we don't have, in order to make this something that's really practical. And now I'm finding that with 3 PhD level people in the lab with faculty involvement, that we're now analyzing, I think, 600 samples from diabetics from Northwestern or University of Chicago, and all these kind of interesting collaborative programs are emerging because they figured out ways to develop assays that are very meaningful for some clinical translational studies. So I think this is one success story that started with the CBC, has kind of had to go through a revitalization process, but now I think it's starting to really work pretty well. I'll mention now some newer areas. One of the small cores that I inherited was really functioned for one PI who left. And I happened to be in cardiovascular research, so I wasn't going to let this die since I had some say in the matter. So cardiovascular research core really was a technician generating heart failure models and doing echo, and then studying this cardiac metabolism, really. And I found that there's a lot of faculty at UIC studying, stroke-related vascular disease, vascular dementia, myocardial infarction-related, cecal ligation puncture, related sepsis and cardiovascular events that follow. So I took this opportunity to develop these survival surgery models, as well as some physiologic recording mechanisms that would be meaningful. So not only the echo, which was funded by a high S10 grant about 4 years ago, but now just skilled people who can do, pulmonary artery catheterization, can do middle cerebral artery catheterization, Doppler in mice that you can now test drugs and knockouts and whatever. And so this is something that is meaningful to me, but I think it's also going to help lead to some significant collaborative and other funding. Connie mentioned the efforts at University of Chicago with mouse or rodent MRI. We also received a NSF grant about 4 or 5 years ago, I think now. Tom Royce and Rich Megan are in bioengineering. They have research interests in elastography, whether it's kidney, liver fibrosis, lung fibrosis, these kinds of things. But the MRI has become, I would say, an emerging but difficult instrument and let's say game to get into. It's not trivial in terms of the expertise to manage it, to run it, to develop new coils to go exactly after what you're interested in, to develop really functional methods as well as the anatomical methods. And so that's something that we're trying to develop. It's evolving. And with that evolvement and recruitment of people like Steve Magno in radiochemistry, that we can now think about doing PET CT imaging. With the School of College of Dentistry, we have a lot of people interested in doing CT, so why not get something like this platform with now the cyclotron available. We can rush chemicals across town. I don't know who's going to be doing that, but I'll help sign them up. And if they have this, what Phil told me, the 6-day half-life of Valadium, so whatever that, Palladium, that maybe that gives us a little bit of leeway. But anyway, so that makes these kinds of things more possible. We just received the shipment of this instrument as a one-year demo, and we're going to start into this game. And ideally, this will generate the data needed to write grants, and ultimately, you know, we have to kind of feed the system initially to get PIs able to use this in generating valid data, and then get a chargeback so that we can recover. In our case, we're trying to recover about 70% of the cost. And so a new instrument, it may take several years before you're able to do that, but I think that's how we have to evolve with the technology and the area. Last two areas I wanted to mention what Greg Thatcher has generated, which is UI Center, collaborative engagement in novel therapeutic research and enterprise. And Greg's vision, I think, is to make this a real collaboration between faculty and the center that has expertise in drug discovery to carry an initial mechanism with some targeted information to the next level. How do you do that? And Greg's developed a team approach to do this with utilizing other aspects of the institutional resources, including the RC cores, where the high throughput screening platform sits, where this BioCore sits, where the expertise in interfacing with purified protein chemists and these kinds of things can all work together so that they can carry out the lead to hit or hit to lead kind of programs. And including actually sourcing some of this out, as someone mentioned, that it's important actually to get secondary views from this. Maybe sourcing out synthesis versus buying a library, and then also maybe on the back end, maybe having some additional collaborative support. So I'm not going to go into this. I think Greg is known in the city, and he's here, so thank goodness. He's had some success with this pipeline discovery. 7 high throughput screen campaigns have been completed. There's a pipeline shift now at UIC actually to oncology as we try to pull things together in concert to support a cancer center designation grant application in about 1.5 years. And the strategic discovery that's going on, I think, is now people are getting more organized in order to target some specific areas that will ultimately, I think, pooling your resources to limit the failures, perhaps. Carries out all these workshops, is involved in training grants, lots of student involvement. So this has been very, very successful. I guess. The second part I'll mention, UIC has had this history of this toxicology research lab for a number of years. Primarily, it's been let's say academic CRO, but tied to NCI contracts and other contracts, BARDA contracts. So they're testing compounds in our animal facility, and this is leading to successes in those contracts, and they're heavily supportive, primarily doing this in this area. Alex Lubomov is the director of the facility, who's kind of grown up through the previous director. He's not here today. He was going to try to be here, but they did submit a contract today at noon, and he said he was spent. So they have some success providing these preclinical services, IND, NDA drug submission, and they've completed over 30 so far. They have really the full gamut of this toxicology program. I'm trying to get him to function a little less as an island and to work a little bit more with me. So, for example, one of the things he really needs is a GLP-compliant histology service, right? So he's sending a lot of samples out. I perhaps can accommodate that, and it will really benefit my core facility. Histology is one of those things that's pretty cheap, $10 a slide maybe, or $10 a sample, but can you do it in a GLP compliant way that will still be fit his bill, in a sense, fit his budgets? So that's one of the things I'm working with Alex on. And finally, I just wanted to thank the CBC. They sponsored some students recently. They went out to a Gordon conference in Boston on drug safety, and one of those students was actually from my lab, Masook, who's on the far left. I don't know if any of the other students are here. There's Masook, but anyway, thank you so much. She'll be happy to share her experience with you. I think it was actually a great thing to do, so thank you.
So why don't everyone join me in thanking the presenters again. So I thought that was a terrific overview, and so what I'd like to do now is turn this into a little bit of a discussion and dialogue and entertain some questions from the audience. But I'll start with the first one. And Phil, you know, I appreciate your teeing up the question of the gaps, right? And I agree. I think there's an awful lot here, but there are still some existing gaps. How do we coordinate the prioritization and the strategy for filling those gaps, because we have so many players in the community? There's clearly a role that perhaps CBC can play, individual cores, individual universities, but there are so many entities, and so I'd appreciate, you know, some thoughts from you on the mechanism to coordinate the strategy for filling these gaps so that we end up doing it in an efficient fashion.
That's a great question. The CBC has had strategies in the past that have worked, and we might want to begin there. For example, in order to take advantage of the cores, they funded 2 different programs on operating costs for cores. One was a program for postdocs to work in core facilities on specific projects. The CBC paid for those postdocs. I think the caveat was the postdoc needed to move into a technique that the postdoc had never done before, and that was a criterion. Clearly, that was something the labs weren't going to do without some help to encourage them not to bear the cost themselves, but they take advantage of the opportunity. That was one. Another was the high throughput, taking advantage of the high throughput facilities at all 3 schools, funds that went to the PIs to fund projects that took advantage of those facilities. So that's 2 examples that the CBC has already invested in the past that were, in my opinion, quite successful because they allowed faculty to do something they wouldn't done before. So taking advantage of that strategy, one approach you might consider is looking around with this kind of group and the group that's here and thinking through whether there is some way we can invest in projects that would take advantage or target the gaps and fund those projects to address those gaps. So that's one strategy. The other is having a meeting like this. So already meeting Stephen, Chad's here, the people at University of Chicago who are running the cyclotron, getting us together to find out the gap is down the street. And so that's just a matter of helping you build opportunities like this to get us together to find out. So that's my quick answer.
I don't have anything to add to that.
Yeah. The only thing I would add is I think one of the gaps that Phil mentioned was mentoring, highly, how do we find we need highly skilled, staff scientists to run a lot of these cores, and I think that's so important, and that it's not just that they're viewed as a core in the true sense of, need something sequenced, you get it back, but that it's really a truly collaborative effort and that there's a lot of knowledge shared and training and education. So I think that's really, really critical to this sort of establishing the whole drug discovery pipeline.
People out there have questions, or you want me to just, I can go on forever as well. I was talking with Gary Schultz and Terry Frederigo at the Lakeside Discovery Kickoff last Friday. And one of the gaps that we were talking about is the amount of medicinal chemistry that we have available. We clearly have, the ChemCore at Northwestern. We've got Greg's shop there. There are, I think Duchasis, I think, is planning on adding some medicinal chemistry. But at least from my perhaps parochial perspective, we're still not impedance matched between the amount of chemistry that we're doing relative to the amount of biology that we can do. Where are there thoughts along, other people share this opinion, or is it just a few of us chemists who are, who are concerned that we're not exploiting? Because some of the points that you raised on your slide about the compound stability and the potency, to me, there's a component of you got to measure and characterize it, but then when you want to fix it, that's a chemistry intensive, and it's an iterative kind of process. Thoughts? I see Dr. Thatcher has a microphone, which is a dangerous thing.
So yeah, I'd like to reiterate what Jim's saying. I think we're at a... That's okay, it's not. So I think we're actually coming to the point where we're going to start discarding HTS campaigns because the quality of the hits might not be quite as good as another one because we don't have the medchem. So what Jim knows from industry is that the medchem resources you have to throw at one successful HTS campaign is probably 10 PhD medicinal chemists per hit. And that's just not feasible. Yeah. So, we've kind of got like one PhD medicinal chemist per HTS campaign, and the more successful you get at the early side, so the more successful you are at having the HTS go through, now you're getting one PhD chemist per 3 HTS campaigns, and it's just a brick wall. And it's not fancy because it's medicinal chemistry, it's not a cryo-EM, it's not something that is really sophisticated and academic, and so it's difficult to kind of see how to overcome that. As Rich showed, we've used a Chinese CRO for medchem, but that costs money. Yeah. And it's efficient. It's really very, very efficient, but the amount of money that you need, and maybe this is where Deerfield comes in the context of Northwestern, but yeah, it's the biggest barrier that certainly we have in terms of moving things forward. Is there a training component to having more PhD medicinal chemists, and they do this as part of their graduate studies? I guess that's a possibility, but then there aren't that many jobs these days for people graduating with a PhD in synthetic medicinal chemistry.
Yep. Other thoughts? Terry, I saw you nodding. I think I'll take that as yes. You wanna comment?
So a couple of ways that Northwestern has tried to add. So we have a chemistry team right now of 7 PhD chemists, and they are funded by a variety of mechanisms, and some of them are on sponsored research because that's where, fundings come in. We've gotten lucky with some grants. Not a lot are on recharge because of the cost of doing the chemistry in the long-term scale of the hit-to-lead optimization stage of drug discovery. What we have found, so first, I should credit a lot of our ChemCore actually started because of a lever award from CBC in joint with UIC and University of Chicago, so thank you. And so with that, we spent our first probably 3 to 5 years actually trying to take a lot of the hits that we had and put them into medicinal chemistry, and we realized it took a lot of education. So I do think that an area that CBC can help us is providing more of those workshops, but understanding CBC, and what we've learned is, or CBC MedChem, what we've learned is the biologist certainly doesn't care how we do our chemistry, but we need to explain to them that it's important that they trust us with their biology so we can do our medicinal chemistry. And that's an area that I really think that, yes, it's always good to get personnel support, but echoing the work that we've done in educating would be very helpful. I feel like, medicinal chemistry, they come look at us like we're just a bunch of dollar signs, and years later you might get that, popular compound like Professor Silverman did, but there's a lot of work. So I think education is a good way to help.
Cool. Thanks, Terry. And I also think that resonates, Phil, with your comment about the mentoring and the guidance as well. And I guess I would argue that that's a role that CBC is interested in playing. I think we're trying to convene a variety of experts from the community and mobilize them. But I think we also believe that even just within CBC, we provide some veterans who can provide that mentoring. Clearly, that's one of the reasons that I came here, is that I've been around the block a couple of times, been involved with a few drug discovery programs, and so I'm certainly would look forward to providing some of that mentoring and trying to help train other potential mentors as well, because I think it is something that we've got to try and amplify.
If that's what comes out of this workshop, this would have been worth just having this night together to be able to recognize, if there is consensus, that building such a program. The University of Chicago, UIC, Northwestern each have little pieces of this that they try to offer for their own. Right. But I think getting the CBC to broker something that doesn't already exist, that is perceived as a gap in this educational piece for the faculty, because clearly the faculty aren't trained to think this way. And I learned this as a postdoc. So my postdoc was at Bell Labs in Murray Hill, New Jersey for 6 years. And I played sports with the guys in the development branch. And what I learned from them about developing products was golden. What they taught me was that anything that people like me in the basic sciences were coming up with, 10 out of the 100 ideas that they would get each year out of the basic sciences had some chance of being useful from a marketing product device point of view for the company. And maybe one of those 100 ideas would hit the market at a time that it would actually be something useful that people would want. And that was very humbling to think that all this great work coming out of the basic sciences at a place like Bell Labs, which had at that point already won 7 Nobel Prizes, and of my peers who were there, 3 more that happened after it all broke up and they went to universities from work that they were doing while we were there. I mean, this is really first-rate basic research, and yet to have the people in the development arm explain to you the odds of anything important that you've come up with being useful to us in making a market. And these were my friends, and I took it to heart that this is a crapshoot.
Right. Well, there's a selection pressure that is clearly pretty strident. Along the lines of education and workshops, I'll put in a plug and get you guys to think about. In the spring, CBC is sponsoring a workshop on a drug safety assessment. It's going to be a full 2-day, or maybe it's a day-and-a-half session. It's going to be taught by a longtime medicinal chemist, but it's really going to go into the details around how does one assess safety risks for a drug discovery program and then mitigate against them. And so it's a former colleague of mine from Lilly, Brian Norman, who's been developing the curriculum for this course in concert with a couple of toxicologists. And so just put a plug in there. We'll be advertising. Nancy's going to be coordinating this course. I think it's going to be in April of 2019. And so I think we are taking this educational mandate to heart, and so I think that's going to be a good one. I think it was a nice compliment. I was really pleased to see the interest in this drug safety assessment Gordon conference, and I think these are the kind of things we'll continue to look for and to try and provide. And in fact, I think Jim Stevens, who was the chair for this Gordon conference, was also being consulted for developing the curriculum for this safety assessment workshop that CBC will sponsor. But if there are other workshops, if there are other areas where we can provide some of this education that is of sufficient interest in the community, please provide me that feedback. I'm certainly interested in that. Question?
Yeah, to follow up on what was being discussed about the kind of tail end of the MedChem situation. We've had maybe 5 students that spend all their time on their research work to develop a compound, and we're at the end of the drug discovery pipeline. So we spend maybe a month doing some in vivo imaging for these students, and they graduate and it's done. So I've seen at least 5 or 6 projects that basically just die because the student has gone on, they've developed this compound, and then the rest of the drug discovery pipeline is left empty.
I have another question about a gap.
Yeah, I'm not sure. I'm not sure that I understand that there's a question in there, but I didn't want to leave your comment just completely unaddressed. I think that is one of the unique challenges of trying to do research in an academic environment, right? While in biotech, we certainly have some volatility in our workforce, there's, this cyclical nature, this transient nature of utilizing the students and the postdocs who are there on a relatively short period of time. I'll need my academic colleagues who have thought about this problem to offer solutions there that are better than mine. But I do think, there does need to be a mechanism in place to at least evaluate these projects that are coming to a conclusion with the graduation of a student or the transition of a postdoc, to evaluate, is that the right conclusion for the project, or is there a mechanism in place to try and, move it on to someone else? So, yeah, microphone's coming.
So, I have a comment. I think when the graduate student goes and the project dies, I think that usually it's because the PI think it's only important for the graduate. That project is only important enough for the graduate to graduate, and therefore he's not pursuing it, or he or she is not pursuing it further. And when it's important, I think they will further pursue it, like we often do. And I think another thing is for that part, to develop a successful drug. I'm not very experienced, but from my research experience, I require deep thought and more intimate collaboration between the different people with different expertise. And I think the CBC here would be great to serve as a dating service for people who get connected to have more intimate, deep collaboration that will generate great products.
Yeah, I actually agree with that. And I do think that we feel part of our role is to help in the matchmaking. And, it's one of the reasons that, I have been spending a fair amount of my time trying to educate myself with the various programs that are ongoing at each of the 3 campuses. And I've been here for 8 months, and I certainly have a heck of a lot to learn, but I feel like I've started to scratch the surface and get some connections. And I think we have some future programs, our Entrepreneurial Fellows program, where we also think we're going to try and use that to cross-fertilize and do a little bit of matchmaking across the institutions. But I do think that's an important role.
I was
Rich.
going to expand a little bit on, I think it was actually in my slide that I got from Greg. One of the gaps I see as a real kind of cell physiologist, cell biologist, is taking a discovery related to mechanism, protein-protein interaction is involved in a mechanism, to now a druggable kind of approach. And I've been advised, I think, primarily by Greg, I'm going to blame everything on him, but you have to have a really robust assay now to screen for. So if you have a protein-protein interaction, that's your key. How do you screen for that? How do you go to the next step to say that if I block this, we could save a lot of people? But developing those robust assays might be a gap. The cell biologists now moving to an alpha screen kind of mechanism, is that something that we can do? Maybe with Greg's help or people like that, is possible. But what does that field look like in terms of going from cell signaling mechanism to an assay that's screenable?
So I think that's a great question, and clearly, clearly I think there are some folks we've now brought into the community. It turns out that one that I know quite well, Laura Bloom, my wife, is helping to do some of that assay development and validation within Greg's shop. But I think we need to do more of that. One of the challenges that I see, I think most of the drug discovery efforts that I see in academia, characteristic of Greg's, are very lean. And I look in industry and I see more robustness. When you were, those of you who went to our symposium last November on small molecule discovery, got to hear from Vicki Rashan from Ribon, and she talked about the flow scheme. And I think they had 9 or 10 different assays where they're measuring, lots of different dimensions of the compound's interaction with the target within biochemical. You're measuring, binding, you're measuring function, you're looking then for a target engagement assay in cells, you're looking for a functional assay in cell that you can then correlate with a phenotypic assay in cell as you scale up. And all of this requires a fair amount of work, because you've got to build those assays and then you've got to utilize those assays and look at multiple compounds. But in exchange for all of that work that has to be done, you get more insights and more certainty around what your compound is doing and how your compound is doing it, so that you can then evaluate in similar kinds of assays what else it's doing in those same cells or in other cells at that same concentration range, so that you can effectively de-risk it to move forward. And it then allows you to ask more demanding, more complex questions of the molecule and interpret the results, again, in the context of what you've already demonstrated. And so, I think there has to be, more of this. And I think, again, I think some of it comes back to resources, that we simply are spreading ourselves thin across more projects than we can handle and can afford. But some of it is just the knowledge and expertise around how you put together, a more complete testing scheme and the benefits. It's not just about filling out a big table, so that you can check a bunch of boxes. It's really about understanding how to ask more, more meaningful questions and learn more from those subsequent experiments.
So, Jim, I see an opportunity here, at least to think this through. What you're describing overlaps somewhat with core facilities and their behavior, we have many of the people in our cores who have backgrounds in industry, who've been attracted to this. The financial model we currently have in CORS, though, is really on the back of the grant-driven investigators who are not risk takers per se. I mean, there is risk in doing the basic science, which we've all done as part of our careers, but it's a different kind of risk. It's one that doesn't cost much other than labor, which we have with graduate students and undergraduates, fairly small labor costs by comparison to industry. But what's missing there is not just the fact that the funding that's driving it is not a match for it, but the costs that Terry mentioned that can be quite substantial to be able to move this forward. So what I'm going to throw out is the idea that maybe what we need to do is put together a group to take a deeper dive, just a thought group, right? This isn't a funding group, this is a thought group that would take an example from a couple of laboratories and think through what would be the cost involved in moving this through a core facility, if a core facility was willing to take on this part of the task. Again, moving beyond what they currently do. For those of you who are here who don't know what core facilities do, what we're talking about, fee-for-service, research, no guarantees, but we have standard operating procedures and equipment and techniques and expertise in our labs that can assure people that if we do the following experiments, we'll get a result under these circumstances. And that's what the faculty are looking for. But those circumstances are not what you described, which is how do we move a product, or an idea, maybe a compound, through a series of tests that are going to lead to a commercialization. And they're not funded to do this, they're not thinking about doing this, and yet there are opportunities here to do it. But if maybe what we can provide for them is a group who can think through from an investigator's point of view, what would be the cost if we were to set up some of our facilities to do this for them? Would it be cost prohibitive? If it's going to be cost prohibitive, then that's not the way to go because there are limited resources. But if it is possible, remember, 30 years ago, 20 years ago, we didn't have core facilities. And if somebody would have said back then, and I've been at Northwestern 31 years as a professor, if somebody would have said back then, we're going to outsource this stuff in my lab, I would have said, you were crazy. Now here we are 30 years later, we outsource about 20% of the research going on in our labs is all done in core facilities now at Northwestern. That's a fairly large number. It's $25 million a year. So it's this cost issue. If maybe that's something we could do, is put our heads together.
Yeah, I mean, I think it's certainly an interesting thought experiment, right? What would it look like? How does it fit the mission? Does it actually meet the needs?
Yeah, because it's really a different model, right? And so to think through how that would work, how it would be funded, I think would be very valuable.
Yeah, I mean, I think there are, clearly in academic drug discovery, which is what we're talking about here, we're at a transitional phase where we are exploring a variety of different models. I'm looking in the audience, I don't see Jeremy Smith. I'd invited Jeremy, who's president of the Rainwater Charitable Foundation. I work with the Rainwater Charitable Foundation as a member of the Scientific Advisory Board for a TAL consortium that they sponsor. And they're trying to advance academic drug discovery. And one of the things that Jeremy and the Rainwaters have funded is an exercise from Milken Consulting to canvas the academic drug discovery models across the U.S. and to evaluate, to interview the various folks from Hopkins, from UCSF, Gladstone, from Vanderbilt, from a whole variety of the drug discovery centers and try and compare and contrast the approaches that they're taking and to really synthesize this into an evaluation and, to give us some guidance around how to build that. And so I was thinking that could be, that certainly could be useful for us here within the Chicago community to benefit. I think we'll probably get interviewed as a part of this as well because, we're doing a fair amount here, but also to be able to see the final report. But we are at a transitional stage. And I think we're all trying to figure out new and creative and more efficient, more effective ways of accomplishing
So
this.
there's one person in the audience here who I know thinks a lot about this, and that's you, Anki. She lives in this world between what we're talking about right now, what the investigators want, which is results that can go into the next grant to keep the research project moving forward. They're not driven or motivated by some commercialization. They're motivated by humanitarian needs. They hope someday, we all hope someday what we're working on will bring benefit to society. But that's different than saying, I want to follow that all the way through to be part of that closing the loop. But Anki lives in this world, both before she came to Northwestern. So I'd be curious how you think, having worked in a core facility now, whether you've given much thought to whether this would be possible for a core like yours to transition into the more commercial, whether it would be cost prohibitive, time prohibitive?
I think it is mostly cost prohibitive. Time, I think, is just building up. Like people mentioned, that you need to collaborate with a lot of expertise, and I'm sure that between Northwestern, UIC, and UFC, we have a lot of expertise there, so we should be able to move forward to commercialization, especially submitting IND application and have that approval and go to clinical trial stage. And of course that we have a lot of physicians in universities that we can work with them to set up the clinical trials and everything. Unfortunately, usually at Northwestern, the pipeline doesn't move too fast from HTA, ChemCore to the preclinical, which is my core. So usually it's back and forth between HTA and ChemCore, but not moving to the next stage. So, and then we're hoping that there would be some funding source that might be able for CBC to select a certain candidate of the drug that can move from, the first step and go for commercialization, which is like basically submitting IND, go to that review with FDA, and then go to the clinical trial. So hopefully the story for Lyrica, it would be repeating.
So I'm somewhere in there, I heard you say that you thought it might be cost prohibitive, but let me ask you in a slightly different context, which is, imagine in a recharge center, core facility, where you created service lines that would, in your opinion, move a project in the commercialization space rather than for what the PI is expecting, which is data to go back into a grant that answers some more narrow question. Do you think it's even conceivable to come up with a cost structure for what would be required to move something in that other direction? You already answered and said, but I want to be clear what you're answering. Are you saying that creating such a service in a facility like yours, which is the diagnostic therapeutic core, is going to be a non-starter?
Well, I guess, there's a lot of PIs at Northwestern right now that has their drug or their peptide, their small compounds are very promising, and I could see that we can put some IND and putting it for, because as you know, drug development is starting with, drug discovery and then preclinical, and then you go IND, and then phase 1, 2, 3, and then you go to the market, right? So the problem right now is usually when you go to our stage, they're always running out of the money, Phil. So, and another trouble is usually when they have to do that GLP tox or PK, which is really necessary for them to get that section to get for IND submission. For example, there's one PI at Northwestern right now, Jack Hankins, who got R24 for creating a peptide that will treat macular degeneration, and that because of lack of, for example, project managerial, meaning not systematically being designed for the whole structure until go for IND application, they're running out of funding. And this is very sad because they got almost $5 million to go from from the beginning, like meaning synthesizing the peptide and selecting that, and then have that candidate and getting it into the door of an IND and go to clinical trial. So at the end, the core can help, but like I said again, our core is not running the GLP. And now, for example, I need to talk to Alex, the person at UIC who might be able to partnering with us to be able to actually work together and have that package at least in pharmacology section or tox and PK so that we can have that section, let's say, covered for IND. We can work, for example, with for CMC section, we can work closely with Terry team that probably they're expert in putting together all those, section, for example. So there's a lot of expertise here. It's just that we have to work together and sit down and think about it and which drug that might be druggable or meaning like have a big potential to go to the market because FDA is going to be so picky. In the past, I work in the really startup companies, so I know the struggle. So usually they're starting to licensing. It's like if they're showing that the drug is safe. So you have to go through phase 1. I mean, it depends on what kind of drug. If the PI can really explain that this is really promising, that might be licensing really early stage in a phase 1, but that's rare. Usually they want to see safe. And you go to phase 2 and showing a little, like indication of a good efficacy, and they're starting to licensing that. So, but like I said again, mostly it's a funding, and I think I'm hoping that working together, that we will be more efficient, and hopefully the cost would be dropping. I'm hoping.
I mean,
so.
To me, drug discovery currently in the current environment. I think there's an abundance of funds, right? You just look, yesterday, 5 IPOs priced, including Joe Moskale's Eptinix. And I think that was the single largest IPO on one day in history. There's an abundance of capital being thrown at drug discovery. The flip side of that is there's a demand for data and the robustness of a data package. And so, my prejudice is that the projects that don't get funding don't have a sufficiency of data or the right data or a completeness, and so to me and maybe it's a little bit of a catch-22 because generating data requires funding, but in general, my experience has been you run into funding shortfalls when you try and jump ahead too far and you outstrip the data that you have. Because the challenge, and I was thinking about this when Greg was talking, when we shortcut medicinal chemistry and try and advance the compound further, we're actually cutting short one of the less expensive phases and moving the molecule into a more expensive phase with still more risk than it should carry. And I think we tend to do that, right? When we become constrained for resources, we try and skip steps and cut forward. And with costs going up at every stage of drug discovery, pretty quickly you get to the expensive stages, and now you're trying to generate larger sums of funding and you've still got issues that you've carried that should have been discharged from the earlier stages. And so I think we've got to think about this from a fundamental standpoint and make sure that at each stage we've got a sufficiently broad and robust data package that it can justify, it can live up to the scrutiny required to get to that next increment of funding. And so I think that's part of the challenge. And clearly that's some of what we're trying to do at CBC is provide both guidance around what those data packages could and should look like, and some level of funding, some level of resource to try and address those. But I think it is it's a challenge, and it's a bit of an insidious challenge, because I think shortages of resources encourage us to try and take shortcuts, which then create funding gaps in the future, because a data package that supports a $250,000 investment isn't equivalent to the data package that supports a $3 million investment. People, want to make that larger investment on the basis of more data, more certainty, more de-risking. And so, I think I think this is an overall challenge. Are there other questions? Mike, you've been waiting for a while.
Yeah, I was going to ask one question, but since we had this big discussion, even another question came to my mind. And that is, certainly you're doing a lot of the great work on campus and in the cores. And one of the potential solutions to what Greg brought up and others are talking about is, and I think Greg's already doing this, is outsourcing some of the work. And certainly the Chinese companies are great, but in my own case, and I didn't mean to make this a marketing message, but our company Shamrock Structures here in town, I mean, we can do medicinal chemistry, we can do cell biology, we can do protein crystallography. Love to work with you guys. What we need is just the funds to pay the people and support the lab. And I'll bet you that when you think about the indirect costs associated with the academic environment, it's probably pretty cost effective. And, because oftentimes we've thought of the academic world as being not able to make the investment, and so we don't seek them out as customers. Whereas we seek out, the biotechs of the world that just did those IPOs yesterday or today. Right. And, but on the other hand, we're actually more interested, I would say, in doing the work with the universities because of the potential that they could lead to, because we also have access to the expertise to do all the things that was just pointed to, preclinical development, into clinical trials, IND filing, all the way through phase 1, phase 2, phase 3. And that experience has been had. So the experience is here, and there are companies that do this. Yep. You have to pay money, yes, but they can accelerate your work quite a lot. That was my point. The only question I have is, I wanted to talk about argon for a minute. University of Chicago manages argon. So is there a way to access argon capabilities under the CBC umbrella, things like the Anjay-Yochimiak Advanced Crystallization Facility, or maybe some of the data science capabilities that they have there? And that was going to be my quick question before you guys went into this big thing about drug development.
So can somebody speak to the argon?
So
you probably know better.
We have two facilities at the Advanced Photon Source. We have a materials science group and a life science group that does nucleic acids and proteins. And again, they're all using the beamline to look at high resolution X-ray crystallography, as you're well aware of, Mike. Those are not set up as recharge. So last year I worked with Steven Streifer, who's the director of the APS, and Suresh, his right-hand person on the finance, to try and think about a recharge model for the APS. The Department of Energy is very interested in this. We don't know where the Department of Energy is headed at the moment. So until this gets hashed out in Washington, it's on hold. But the answer to your question is that at universities, at least throughout the world, there are universities who have access to the beamlines through their own collaborative access teams. In most cases, some companies have their own access to beamlines as well out at Argonne. So if your question is, how do you find access? I think there are all kinds of possible ways in which a company that's doing research with a compound can gain access. It's just a matter of cost. Everybody else is paying, that's no different. But the recharge model, the interesting thing about recharge model at our facilities is we'll take anybody. And Argonne's not set up that way at the moment. But they are interested in moving that direction, but my suspicion is it could be several more years. What they're, as you know, they're planning to upgrade the beam, and as part of that upgrade, they are open to the possibility and are consulting with groups, including myself, about how might that look if we were to convert this to a recharge model.
So Julian, maybe I'll let yours be the last question, because I think we do have some cocktails and some snacks waiting, and maybe people would like to take advantage of that. But I know you've been waiting for a while.
Well, I don't want to keep us from goodies. Earlier, we were talking about, well, we should select the projects that are most likely to succeed and get behind those. And that's how companies work. Universities are a collection of lots of small businesses, and small business A has very little interest in giving up small business A's interest and supporting small business B's project. And so I think a way, to the extent that we can figure out how to tackle that kind of issue as it relates to some projects that maybe are better collectively supported than individually small business supported. Now, is that in the realm of CBC? I think that's a superset of CBC and into the realm of deans at the universities.
So I think that's a fantastic question, and I have some experience in that area. When I joined Eli Lilly, Lilly operated in a very investigator-driven fashion back in the late 80s. Every senior scientist was considered a PI. And one of the challenges that we had was that the incentive structure was such that you did your own individual research, and we didn't have mechanisms in place to marshal forces and do something that required more than we could accomplish within our own lab, where it really required more of a consortium of investigators. And so, and I think we explored a number of mechanisms to try and get people to work together, but it's a difficult problem because, I also think that the academic mission is crucial, and we can't lose sight of that, right? The reason that universities exist to train, the educational part and the basic science part, and that universities don't primarily exist to create commercial opportunity. It's that there is commercial opportunity that comes as a natural outgrowth of the fundamental mission that the university supports, right? And so how do we walk that line? And so, at least in part, one of the things that we think about is the investigators are great sources of initial ideas. And, is there a way of allowing them to pursue their dreams and ambitions and broad ideas, and for them to nominate at least a subset of those ideas to move into more of a commercial machinery, if you will, that can take it forward. I remember having this conversation on several occasions with Greg Verdine over the course of his career. I got to know Greg in the early 90s when we made him a Lilly grantee. And he would say, I need to go back at what I'm good at as a Harvard professor, coming up with crazy, insane ideas that have some potential, but I'm not great at turning them into companies or commercial opportunities. And so how do we find a way where I can be the idea generator, and then there are other people who are capable of taking those ideas and translating them? You know, I think this is a question that goes probably beyond my intellect and my pay grade, but those are some thoughts that I have.
I'm going to be respectful and not ask a question like you said, but I want to make a comment before we close up. Back to what you said about the funding, and the universities are not intended to do commercialization. I want to be clear, as I'm representing INVO, that actually the intent of the federal government is to try to get some commercialization assets out. If you guys discover fantastic drugs that never make it to the public, that's against their intent. And based on the Biodole Act that was passed in 1980, the whole purpose of what they intend to do now, and that's why they're giving universities the licenses to the IP. When funding comes in for R01s and research and everything, and you make a discovery, that discovery then is given as a license to the university. And our mission as a tech transfer office is to get that out into a commercialized product, or create a startup, or create economic growth, so that that money can come back into the government, and then the R01 cycle and gets continued. So it is the intent of the government to try to commercialize some things out of the universities. You can still ask my question.
She was the first one who had a question, so if you make it quick, everybody's getting hungry. Yeah.
Yep, please, go ahead.
So actually, it's more of a thought-provoking question. As you guys talk about different HTA libraries, and part of my tech transfer role is to ask you, I don't know where these libraries come from. I don't know who owns the composition of matter, but there is a great opportunity here. We have a ton of researchers that are doing target discovery. There's a lot of different targets that are being discovered. We know that there's places where you can actually find molecules to address those targets. Is there a way or a model that you can think of that we can pool HTA libraries, get more HTA libraries, as long as we've been very clear who owns the IP behind those libraries? So it's a very maybe thought-provoking question. We can discuss it next time. Or
Yep. So I'll put one comment on that. Most HTA libraries are not comprised of novel molecules. The vast majority of compounds in screening decks are not patentable specifically for composition of matter. There's an awful lot of, many of them are commercially available, and so they are in the public domain. There are often some small fractions of those libraries that are, that are in fact novel, where they come, through supplementation coming out of the chemistry labs. What's novel and potentially patentable is the intersection of that chemistry with a biological endpoint. But typically, those patents aren't worth that much. It's much more worthwhile to patent the molecule that's ultimately going to move into the clinic, as opposed to trying to patent the lead, where all you're going to get is a use patent on a compound that's already in the public domain.
So we're planning a workshop, one of our annual workshops, and the title is going to be, I gave you a target, what do you mean you can't give me a drug?
So maybe we'll let that as the last word, and let's reconvene over in the social area. But before we do, thank our panelists again for, I think, a very stimulating discussion.